Yeah, thanks. So, as you may have seen from the program, my slot was quite empty in terms of the description of what I was going to speak about, and that's because it's a new assay that we're sort of announcing today. We've been working on this over the last few months. It's called ProStorm, and it's basically a cytokine release assay to look at cytokine responses. So, just an introduction to the sort of phenomenon of cytokine storm. Many of you will be quite familiar with it. So it's the issue of potential first infusion reactions that occur in response to biotherapeutics, and they can occur very quickly, within minutes. It can be quite mode of action dependent, depending on what your molecule of interest is actually doing. For example, triggering an immune receptor, kill cells, or tumor lysis syndromes. So the symptoms can range from mild to fatal, and that's really the problem. And they include a variety of potential symptoms, but clearly potentially some very severe reactions. And everyone, I think, is familiar with this worst case example, I guess, to date in a phase 1 trial of TGN-1412, which caused really severe reactions in all test subjects within minutes. And although nobody died, all of the subjects had severe responses and also continued problems afterwards. So first infusion reactions occur in various biological drugs to a varying degree in terms of incidence, and also they vary considerably in severity. We've already heard a little bit about Campath, which causes often first infusion reactions. The TGN-1412 obviously caused a very severe first infusion reaction, but also the other biological drugs that are sort of quite familiar there, Remicade, Humira, Herceptin, Rituxan. All of them have some degree of first infusion reactions, although in many cases they're not as severe as certainly in TGN-1412. So I think in 2009, when we were not really in the game of immunogenicity prediction or analysis, the EMEA held a workshop on this after this Tigginero debacle on TGN-1412 in 2006. And from literature, it looked that there was a sort of white paper that came out from it that was published in Cytokine. So that involved a discussion on improved assays that can potentially deal with this issue of cytokine storm. I dare not say predict, because that's very difficult, but certainly kind of give more information as to the potential hazard of this phenomenon occurring. And the participants generally agreed that it would be obviously very nice to have an in vitro assay that kind of can deal with this question. And that it's probably, however, not really possible to develop an assay that can fully quantify the risk. But in any event, the cytokine release syndrome is usually linked across multiple cytokines, so therefore it's also good to consider the release or measure the release of more than one cytokine, certainly. And what wasn't agreed was what format of an assay should be developed, whether it should use whole blood or PBMCs and the like, exactly what controls should be used, how the data should be analyzed, and of course it wasn't clear how predictive the assay would be. So we've had a stab at this, partially also inspired by what Anja Langenkamp presented here in this meeting last year, which dealt with this question. So the objective is to design an assay that specifically indicates when a cytokine first infusion reaction may be a risk. And so to keep things simple, this assay is based on fresh whole blood rather than using PBMCs or freezing them or storing them. Because it seemed to make sense, and this concurs with what Anja said, that the less you manipulate your sample, the better a chance you potentially have to get a measurement that's hopefully accurate and relevant. So drugs are then added to fresh whole blood in a dose escalation range, which covers the envisaged exposure level in patients. And as an example, that could range typically from 0.1 microgram per mil to 100 microgram per mil. But of course, it may be drug dependent or indication dependent, and depends on, it's particularly the program that you're looking at. And again, we would carry out this assay on 20 to 50 donors typically. In this case, rather than working from our own donor bank, which we have in place for some of these other T-cell assay studies that various people have talked about, these donors are actually recruited for the program, for this study specifically by ourselves, to give blood samples, and we then test those samples fresh. Usually we would propose using healthy donors, but there's no reason why we couldn't recruit disease state donors as well. It's just a question of how likely it would be that we can complete the actual enrollment, depending on how esoteric the disease might be. And then basically once we have collected the blood and administered the drugs, then we collect the plasma and measure the cytokine levels, and we use the ProRay Ultra platform for doing this. So a little bit more detail about the assay. So as recommended by this workshop, we look at a range of cytokines. I think the ones that are most indicated for looking at cytokine release syndrome are actually IL-6, IL-8, and TNF-alpha and interferon gamma. But also we look more sort of for completeness at IL-2, IL-4, and IL-10. And then we measure this in this assay against some well-established comparators, and although Erbitux is potentially high immunogenicity in terms of the antibodies developed in terms of first infusion reaction, it's actually usually relatively low, with a few exceptions, as you'll see. And PBS is also our baseline for this assay. SEB is simply a positive to make sure that the response can be elicited. After all, it's not particularly meaningful, of course, because it's so unlike any biological drug. And CAMPATH is a high-risk comparator. So although the severity of the CAMPATH reactions are not as bad as TG and 1412, they occur with a very high incidence, and so that's a nice one to use for that purpose. And of course, we would always recommend for the sponsor to include their own comparators. There's nothing to say that these are really the best comparators. These are just some that can be chosen, on a routine basis. But it's certainly advisable to include your own comparators. And it would typically, and has taken us 3 to 4 weeks to recruit donors. And carrying out the study takes another 3 to 4 weeks, depending on the study size. So 6 to 8 weeks is a full study turnaround, and for producing data on 20 to 50 donors. And as suggested by the EMEA workshop guidelines, we look at absolute and relative level changes of cytokine release, and also we look, of course, at multiple cytokines. So our donor program, in summary, that we've developed specifically for this assay, but which we could also use for other assays. This is how happy they were once they got paid for donating. But basically, we can recruit them for a fresh blood donation. They register via our website. This is outside of our normal PBMC processing. It's relatively quick, and again, the donors can have a profile that's desirable for the sponsor. And here, in contrast to our PBMC bank, we also can potentially go back to the same donors if they agree, and do further study in the future if any relevant data sort of come up. So in terms of workflow, again, hopefully I'm not repeating myself too much, but after the recruitment, we take obviously the blood in anticoagulant tubes. We then plate out for this assay and add the test items at all these concentrations and the controls. We incubate for a predetermined amount of time, separate the plasma, and then do this multi-cytokine analysis on ProRe Ultra and write that up in a report. Just as a recap on ProRe Ultra, we've adapted this platform to measure cytokine responses. It's a high-content peptide protein microarray, can deal with relatively low sample volume, and has a high feature number. And we typically run the analysis in 24 subarrays. And the point is that the signal performance is generally superior to ELISA, we have qualified this technology basically for a sort of multiplex cytokine analysis to be able to use it for this purpose. This is some unrelated data sort of on some epitope mapping we've obtained in comparison with MSD. So we have a similar comparable linear range in the assay, and also low CVs, but specifically for cytokine detection, we're achieving a detection limit of about 5 to 10 picograms per mil. So in an optimized setting, the detection levels are very good for this platform. So then I'll show you some data for an example study. So we've recruited 30 donors for this study and then tested a number of articles, including Remicade, aggregated Remicade, Erbitux, Campath, Herceptin, and Vectibix. The aggregated Remicade results are quite interesting, and certainly, as you'll see, are sort of going to be an impetus for doing more work on this. It was sort of a last-minute decision. Oh, why don't you stir this Remicade a little bit overnight and let's put it in and see what happens. So we didn't particularly pre-plan that or characterize what happened to the aggregated Remicade. So the bioanalytics isn't complete by any means. So the data has to be taken with a bit of a pinch of salt in that regard. PBS was used as a negative control and also to remove outliers, because you do occasionally see significant responses in PBS. And SEB is used as the overriding positive Th1 control. And then again, Erbitux is used as a drug negative sort of control that has low incidence of first infusion reactions, unless potentially you have this kind of hypersensitivity reaction, which, well, we don't know whether any of our donors had that particular reaction. Campath, of course, again, is the positive comparator. So after we removed the PBS outliers and the PBS background, we had 29 donors that passed in the final analysis. And as you'll see, one of the sort of potential cutoff gates that we've drawn, and in line with what Anya has done, is to do this 95th percentile threshold, meaning that really you're hoping that, or basically you're characterizing that 95% of Erbitux responders should count as a sort of negative response level. Whether that's the right cutoff or not is debatable, and you'll see the issue is a little bit still the number of donors being only around 30. It would probably be more relevant if you had a few hundred, but it's the same fundamental challenge here as with T-cell assay, to design a reasonably sized study and then doing good statistics on it still. And certainly we would concur with the idea that the overall analysis has to incorporate absolute and relative measures of the responses that you're seeing. And the cut points will depend on quite a lot of things, but certainly also be drug and dose dependent. So I'll slowly take you through some of these result data. So what you can see here is, first of all, categorized by the drug that we put into the assay. This is all for IL-6. Each of these boxes is, I guess, a different concentration of the drug, and the cytokine level in picograms per mil is plotted on a logarithmic scale from up to, I guess, 100,000 picograms per mil on this scale. And the donors are always lined up in the same order. So if you see, the same donor being moved off in this sequence off the baseline, and that would be the same responder. And this 95th percentile of the Erbitux response is drawn in with the red line. So that typically means that one of the Erbitux donors is sort of considered, that's the threshold to the positive level, potentially if you consider that sort of as a cut point element. But certainly, as you'll see, it's also very relevant to look at absolute levels and how many of these responders sort of move off the baseline, as it were, at least above sort of 10 to 50 picograms per mil on some of the cytokines. The levels that are significant also vary by cytokine. Certainly IL-6 produces a much higher absolute amount response than some of the other cytokines like IL-2, interferon gamma, and so on. So what you can see is that with Erbitux, really there aren't all that many responses. With Campath, pretty much everything moves off the baseline for all concentrations. What's also not uncommon is what you're seeing for, I guess you see it more on the plots that are to come. Remicade has a sort of declining response. Actually, the lowest concentration is the one that stimulates the most significant response, and there may be some sort of exhaustion phenomenon associated with this. In the case of Campath, it may simply be that Campath, you see it in the context of Campath as well, that the higher doses sometimes give a lower response, and that may simply be that the drug has killed so many cells that there are not enough cells left to continue to respond. What was certainly surprising is this effect with aggregated Remicade. And again, the concentration that's sort of put out here is the concentration of starting material before aggregation. So we can't say that this was the accurate final concentration of this compound. And also, when we put it over size exclusion column, nothing came back. I think all of the material was actually bigger than the void range that you would still see on size exclusion. And this was stirred for 24 hours at 700 RPM. So it's simply stir stressed, and again, perhaps re-emphasizes that shaking or stirring is potentially much worse than heating things up. Yeah, so certainly that was a very surprising result, and I can't tell you much more about what this substance was in the end, but certainly we're curious to find out more over time. So other products like Herceptin and Vectibix do seem to fall into the sort of intermediate range of having some responses, also some at a considerable level, but generally there's clearly a lower response rate than with Campath, and a higher response rate than with Erbitux. On the other cytokines, you really sort of see a similar profile. Here you see a more significant kind of decrease, I guess, at least in the number of responders on Remicade over the concentration range. You see all people responding to Campath, and in this case an increase over the range of the overall response level. And again, sort of low but somewhat mixed picture with Erbitux, and again Herceptin, Vectibix are sort of in between. And the sort of trigger effect of this aggregated Remicade is sort of more profound on IL-8 perhaps than IL-6. Interferon gamma is a when you compare this scenario is very different. So we don't see a lot of gamma release, essentially almost nothing on Erbitux, but Campath, and this correlates with Anya's data, does have a consistent interferon gamma response, and this sort of again this declining response on the Remicade samples, and some intermediate levels on the other two. And essentially a similar picture on TNF alpha with some subtle differences, and at this stage I can't profess that I can explain to you what exactly the fine differences are, but it's quite a sort of rich data set allowing sort of further analysis on what is seen. This is a slightly different presentation of the results in a more linear graph, and also we've plotted some of the SEB data, which very reliably gives a high response here. And again, you see the strong IL-6 and IL-8 responses with the aggregated Remicade, and then also Campath, I guess, is quite clearly distinguishable from baseline in this assay across all concentrations. You see much less on IL-2 and IL-4, and you see some interesting responses, and these tend to be the same donors, and certainly one can do more statistics on these responses, and that's something that we're in the process of doing to see, whether that's has got any sort of specific statistical meaningfulness. I would say that one thing that we noticed was you see also a strong IL-10 response on this aggregated Remicade result, but actually, although you can't see it here in this graph particularly, those donors that are sort of lighting up here as a IL-10 high responder showed lower responses on the other cytokines, like IL-6 and IL-8, I believe, in this assay. So that there does seem to be this inverse correlation of IL-10 with other cytokines, as it's known to happen. Again, a slightly different graph of mean responses, and this scale is slightly suppressed by the aggregated Remicade result. So there are things happening that you can't see particularly well on IL-6 and IL-8, but it's sort of dominated by this particular result. And again, you can see the specific concentration-dependent decrease in some cases of the response for Remicade and Campath. For TNF-alpha, and also here for Remicade, this point is a bit of an outlier, but the general trend is sort of similar. So when you analyze the data overall with several objectives, or compare several analysis objectives, I guess you can see a general correlation between the clinically observed responses and what you're seeing from this assay, especially when you're looking at these typical cytokines for cytokine storm of IL-6, IL-8, interferon gamma, and TNF-alpha. So overall, Campath consistently comes out as a high responder, as is true in the clinic, and Erbitux is a low responder, as is also true in the clinic. And the other drugs lie in between, and of course the aggregated Remicade is an entity that behaves very strangely, but certainly needs to be analyzed in a more controlled way in the future. So generally, the assay helps you to give you a flavor where your drug might lie. Again, we don't say that this is a necessarily predictive assay. It has some of the similar problems as were discussed for the other assay types today, but fundamentally there is the issue that somehow you need to identify potential hazards for cytokine release at a preclinical stage, and we think that this is a good way forward to make that analysis, at least in cases where there is significant risk potentially. There are many caveats, and I've talked to some of them. Certainly, how significant you assume the risk is for a first infusion reaction being problematic has to be taken in the context of the risk-benefit potential for your program overall. Of course, you need to think about how big the cohort needs to be to achieve sufficient study power, what dose range may be really the right way of applying this in. Certainly we would suggest putting in more comparators than we have used in this study necessarily, and different ones will be applicable to different programs. And again, the statistics used to kind of analyze these data is also very much driven by the particular study objective that you're after. So we can help with that. We're developing our statistical analysis capability in this area, but yes, it will be a sort of interaction with the sponsor to decide what's the best way forward. And yes, it can only be regarded as a guide for hazard identification at this stage, and it's certainly we've not, it's not a validated assay in terms of its predictiveness. And also, I should say, it really only predicts these first infusion reactions, which are type 2 hypersensitivity like. It certainly doesn't predict any other hypersensitivity reactions like type 1, 3, and 4, which do require prior sensitization, which isn't achievable in this healthy donor set. So risk quantification, therefore, is not possible, so we don't want to oversell it in that regard. But at the same time, equally, you can't regard the assay result as a go, no-go gate, because that's the other question. So what if it looks bad? Does that mean I can't continue? Really, you should regard it as a tool for hazard identification. Is there not a potential problem? And if it looks like there may well be a problem, then that may impact the dose escalation for the study that you might want to consider. And also, of course, patients can be pre-treated to ameliorate the symptoms of potential cytokine release. So in summary, ProStorm is a cytokine release assay for hazard identification and risk management. On the compounds that we've tested so far, the response behavior that we've seen seems to correlate with what is observed in the clinic, sort of in generalized terms. It's what I think is appealing about it is it's a simple whole blood assay that minimizes test system interference, and we can deploy it in a flexible study design based on a cohort that we specifically recruit for the project and on the sponsor's objectives. And also, as always, we try to deliver this with a reasonable turnaround time and cost. So that's it. And I should acknowledge the R&D team at ProImmune who've worked hard to make this assay happen relatively quickly, and everyone else who's had an input into this. Any questions?